The overall goal of this protocol is to express and purify recombinantly derived elastin-like proteins for use as a tunable hydrogel for 3D cell encapsulation. Additionally, this protocol presents the methodology for downstream fluorescent labeling and confocal microscopy of encapsulated cells.
This method can help answer key questions within the fields of biomaterials, regenerative medicine, and cell biology, such as understanding the role of cell-extracellular matrix interactions in 3D.
The main advantage of this technique is that it provides a reproducible, tunable, and modular material platform that is amenable to the study of multiple cell types.
Overall, the implementation of this technique can improve our understanding of the critical cell ECM signaling parameters that may dictate the success of future cell-based regenerative therapies.
Streak an ampicillin and chloramphenicol agar plate with a pre-made bacterial stock containing a vector that encodes for the ELP of interest. Then incubate the streaked plate upside down at 37 degrees Celsius overnight to allow for bacterial growth. The following day, pick a single colony from the plate and inoculate the pre-warmed starter culture. Incubate the culture in a shaker at 37 degrees Celsius for 16 hours. After the incubation period, use a serological pipette to transfer 20 milliliters of the starter culture to each expression flask. Leave the expression flask shaking for 1 hour. After 1 hour, measure the optical density at 600 nanometers of an aliquot from the expression flask. As soon as the reading reaches 0.6, reduce the temperature of the shaker to